Without the notes. Okay, so I'm going to talk about the Real Vegan Cheese Project. I'm pretty excited about this. I actually had this idea independently of the rest of the group a little over a year ago, or a year and a half maybe, and then found out that several other people in the group had had the idea years earlier than me. So it's a good group of people to work with. Many of us independently came up with the same idea, so there's a lot of energy in the group. The whole presentation is, do you have a pointer by any chance? A laser pointer or anything? Okay, I don't really need one, the screen's low enough. So this whole presentation is Creative Commons licensed if anybody wants to use anything from this presentation. And I try to give attributions for the images where I can. Some things I didn't actually know where they came from. So I'm going to try to play this video. It's a really good introduction to the project. I'll go over the same stuff again later in more detail, but it's a pretty good place to get started. Except for we seem to have, oh yeah. We have no audio, so. It seems to be coming out of my laptop instead, so. I can skip the video. I wanted to show it mostly because it's actually pretty funny. We did a good job. This was the video we did for the Indiegogo campaign. So if any of you saw us on Indiegogo last summer, this was the video. I was going to go over all the same information again. I just don't have quite as high production value as we had for this video. So I'll go ahead and skip this. So we started this project with an Indiegogo campaign to fund it. And so we did this last summer. It ran for 2 months. Our initial goal was $15,000. We raised $37,000. We had, and these stats are from last October. So of course the campaign's been over for a while, so we don't have a lot more related to this campaign, but we had quite a bit of engagement. We had a lot of people talking about this, and not just the vegan community. We had a pretty wide-ranging engagement with this. So this is the team. This was before I joined, so this was about this time last year, probably about a few weeks before I joined. We have a really wide range of people working on this. Some of the people like Patrick here, who some of you may have seen speak before actually, he has a PhD in biology. He's a computational biologist. We've got a couple other people here with formal biology backgrounds, but a lot of these people are computer programmers. We have some high school students. We have people that have all kinds of different backgrounds, and an important thing here is that we're all volunteers. No one's paid for this project. We do this on nights and weekends. So keep that in mind when I show you the progress stuff, which if you all read the Wired article, you might think that any day now there's going to be a product coming out. We're actually not quite that far along, but that's how Wired articles always are, right? They always make it look like this is a thing that's happening right now, where really, yeah, the project's going well, but it's a multi-year project and we're 1 year in. So this is where these things happen. So Counterculture Labs is in the Omni Commons building. It's a collective of collectives. Everything is run as a non-hierarchical organization, including the building itself is an organization made up by all the other organizations in it. It's in Oakland, in Temescal, so not too far away from here, not too hard to get to. We're also working at BioCurious, which is in Sunnyvale. And I should point out that Counterculture Labs right now is running a Kickstarter. So if anybody wants to help us speed this up, we're trying to do plumbing and electrical work so that we can do more biosafety level 2 stuff, which actually I don't go into later, but we're trying to build out the lab a little bit better right now. And so the other people that work on this would kill me if I didn't put in a plug for the Kickstarter, so I just did that. So why are we doing this? Most of the people in that picture that I showed are not vegan. There's only a few vegans working on the project. I'm one of the few. We actually started this for many different reasons. So one thing is that the whole point of Counterculture Labs and BioCurious is to give people a chance to work on these biotechnology projects, even if they don't have a biotech background. So this is a way that anybody can come learn these skills, because maybe you can't go to grad school, or for some reason it doesn't make sense to you, or you just don't want to, but you want to learn some of these things. We're a place that you can come do that. We also wanted to show with this particular project that community labs can do something on par with university labs or large corporations, something as important and relevant as high tech. We also want to change public perception of genetically modified organisms. A lot of people don't like them, and I'm going to talk about that later. Yeah, I'll get into that later. But of course, the environmental impact of agriculture, a lot of the non-vegans in the group still see that as a major goal. In fact, everybody in the group believes in that point. And then, the vegans in the group and some others who are not yet vegan would like to see the end of using animals for milk. And so this is my personal perspective, is that this is not about making better cheese for vegans. This is making a vegan cheese that everybody else will accept. And so I'll get into why that could be true in this project. So I won't spend too much time on this. Probably most of you know some of these things already. But dairy is a pretty significant part of the world's greenhouse gas emissions. It takes a lot of water to make milk, 10 times more than almond milk, which you've probably read about the impacts of almond milk recently. Other things, this was kind of a nice graph. It's in weird units. It's equivalent CO2 per dollar spent on that food, but it still shows the difference between different kinds of foods very well. Red meat's the worst for CO2 emissions. Dairy is close behind. Interestingly, if you look at that, we should all be eating in restaurants, ordering other miscellaneous food and beverages, which actually describes my diet pretty well, so I like this graph. So you've probably all heard, I can't go vegan because I like cheese too much. I think everybody's heard that before. I actually tried to find some sociology studies on this or something. I couldn't find any academic research on this, but the idea is if we have a vegan cheese that is identical, as far as anybody can tell when they're tasting it, if it feels the same, if it stretches the same, if it seems like the same stuff, people won't care if it's vegan if it's like regular cheese and cost competitive. So this will take away this excuse. So what actually makes cheese cheese? And it turns out all the things are important. So if we look at the sugar, you need the sugars in there, but this is the easiest part to replace. You can use lots of different sugars and get something very similar. The fats, those are important for those of you who at one point ate cheese or maybe still do. If you've ever had low-fat cheese versus full-fat cheese, there's a big difference. But again, the fats, there are certain fractions of like coconut oil and some other oils that act very similar. The fats in dairy are nothing really that special. So what's really important here is the protein. It is one protein, casein, is 80% of the protein in milk that gives cheese all of its physical properties, the meltability and stretchiness, and a lot of the flavor as well. So what is casein? Why is this stuff hard to make? So it's actually a complex of 4 different proteins. The two alphas and the beta are completely inside this big ball of protein, and they're held together by calcium nanoclusters. And they attach to the calcium nanoclusters with phosphate groups. And so they want to separate out. They're hydrophobic. They want to separate out from the water. But then this kappa casein has one end that's hydrophobic that wants to stick into this ball, and one end that's hydrophilic that likes the water. So it keeps this whole thing suspended in solution. So you can see it's a pretty complex structure. Those phosphate groups I mentioned are put onto the protein after the protein is synthesized. So this is called a post-translational modification. So there's another step where you put phosphorus on after the protein's made. Same thing happens with sugars on the outside of the kappa casein that make it more soluble. Interestingly, even though it's this complex structure with all these steps when it's made, if you take the kappa casein and didn't have sugars on it, it still more or less works. It's not quite as soluble, but you get something similar. So you might be able to get away with not having those sugars. And it looks like maybe the phosphate groups are not 100% needed. We're less sure about that one. So how does this become cheese now? So we've got, this is a picture of the same thing I just described, this big ball of protein with calcium in it. There's actually some fat in there too that gets sucked in. There's this enzyme called chymosin. This is the most important enzyme in a complex called rennet, which you may have heard of in cheese making. And it cuts off these capsaicin tails, the part that keep everything suspended in water. So all of a sudden, it's no longer soluble in water. And so they start to collide with each other, and they make this whole three-dimensional network that's the cheese curd. And so this network, each individual piece is pretty flexible and stretchy, but they are held together with pretty strong nodes. And so you can pull this thing, and it stretches, and when you let go, it springs back into shape. Those nodes are weak enough, though, that when you heat it up, they will melt and flow. But then when you cool it down, these networks reform. Pretty highly specialized function here. So could we find something that acts like casein from other sources? There's a lot of people doing this kind of thing right now. A lot of really good work being done in making vegan foods that act the same completely from plant sources. Well, this one's particularly hard. It's a pretty specialized solution to a problem. Young mammals take in a lot of nutrients, just like all young animals do, but mammals have this specialized solution of milk, which allows you to have more calcium, more protein, more fat, and more of several other nutrients than you could possibly dissolve on their own in water. So this is a really efficient delivery mechanism. So you might be able to look back into evolutionary history, find a plant that diverged from a common ancestor and find some protein that's sort of similar, but no one's found that yet. There doesn't really seem to be anything that similar to casein. So if we really want casein, if we want something that's really acts the same, the best way, at least the most direct way, is to just genetically modify an organism to produce it. And that's what we're trying to do. So the obvious question is, well, you're using animal genes. Is this really vegan? And there's a chance that some of you will disagree with this, actually. Hopefully not. But here's kind of the basic idea of the information, what we're doing. So we're reading this genetic code out of a database. So that's where we start, is we start with information stored on a hard drive somewhere. And use that data to synthesize DNA from non-animal starting materials, and then get this DNA into the yeast. We can grow it on vegan media, and then we get this protein out. So really this all is probably, I think most people agree that this is vegan. The one step, somewhere along the way, we had to get this data from an animal. So cows were probably not treated this nicely, but you can get it from a saliva sample. If any of you have ever done 23andMe or one of those similar things, it can be a very non-invasive technique. In fact, I'll talk about narwhals later, and narwhals, people track narwhals, they're endangered. So when divers from UC Santa Cruz were going to do health checkups on narwhals, they take little skin samples and can sequence the genome from that. So this is the one place there was an animal involved at one point, but past that, I think people would agree after that's vegan. That's the point you can argue about. So why are we doing this with yeast? That's the organism that we're going to introduce this DNA into. And really the answer is that if you look at the tree of life, the fungi are very close to animals. Bacteria, which also get used in genetic engineering a lot, are much more different from animals than fungi are. They have a lot of the same molecular machinery that we do. And one important thing, I mentioned that there are sugars on the outside of these micelles. Well, yeast put those sugars onto proteins already, and they do it in a very similar way to the way we do it. So if we're lucky, we don't have to engineer that in. They might just do it. We're not sure. We don't know this part for sure yet, but there's a chance that we don't have to engineer that step in. So I just told you that it's basically data from a database that we're putting in. How do you get that data in? Unfortunately, it's not quite that easy, but it's not as hard as a lot of people think. Any of you could learn to do this pretty easily. In fact, if you want to come work in a lab, we'll teach you how to do this very, very easily. Okay, so we're using plasmids. And so plasmids are little circular pieces of DNA. Well, this is a definition from Wikipedia. It's a piece that is not integrated into the chromosomal DNA, what you all think of as an organism's DNA, but it still gets replicated inside the cell, and the proteins still get expressed. And yeast particularly take up plasmids well. So I'm not going to go through all these things. In fact, I'm a synthetic chemist. I'm not a biologist. So I could tell you vaguely what some of this other stuff is, but I couldn't actually tell you all of this stuff anyway. There's a lot of other instructions here. So everything here, we didn't invent any of this. This was an open-sourced plasmid piece that we used. We're open sourcing all of our work as well, so anybody can use this whole plasmid. The part we did design is this part, and there's two really important things there. There's the casein or kinase gene. So the kinase, when I said there are phosphate groups, the things that hold it all together in the middle. We're making some of those because we think the yeast may do it, but that's less likely that the yeast will do that well. So we're expressing several casein proteins, and then we're expressing some kinases. There's one other really important thing here, which is the alpha factor. So this is a little tag that goes on the end of the protein when it's expressed, and this tells the yeast that when it's done with the synthesis, to kick it out of the cell, so it's just floating around. This will make it a lot easier to purify later. And that alpha factor gets cut off, so you won't be eating that later. So it gets out of the cell, gets cut off, gives you your protein. Very convenient thing to have. So there's a couple other things about getting it in. Basically, the yeast, sure, it fights back. It doesn't want any DNA to come in. We can add some chemicals and get it to go. One of the weird things is that yeast, pretty much all cells have defenses against DNA coming in, but you can flood those defenses out. For some reason that no one's been able to explain to me, the standard flood DNA that you use, the carrier DNA, is salmon sperm. No idea why. To keep it vegan, we're using strawberry instead, or we're transitioning to strawberry DNA right now. So I made my little graphic there. The interesting thing about strawberries is that they have a really high portion of DNA. That's like things that you normally eat. There's more DNA in strawberries than anything else. They've got 8 copies of DNA where most organisms have 2. So, and there's some interesting evolutionary biology in there too. So once we have these organisms that are engineered, basically what we do is we feed it sugar. It spits out protein. So there is some input here. Sugar is not always a great, sugar cane can be a fairly environmentally impactful thing to grow. It's not as bad as meat, though, not as bad as dairy, and really any sugars. You could find alternative sources, but we do need to feed it sugar. We're hoping that we get the native yeast enzymes to add the phosphates and the sugars. We may have to engineer some of that in, may not. We should just be able to separate out by filtration. There's a chance that we'll have to do some fancier things that will be more expensive. We're hoping not. And if the native yeast enzymes don't work, we'll have to then maybe add our engineered enzymes later. But then what we're doing is each of these individual sections, I said that there are multiple different proteins that came together to make the casein micelle. We're growing each of these in different strains of yeast, purifying them and reconstructing them. This has been done before, though, not with yeast, but taking the bovine, the stuff out of milk, breaking it all down into its parts, and then mixing it back together works. We already know that this step works. So this part should not be a barrier, unless we have done the phosphorylation or sugar adding. If we haven't done that well, it may be hard, but if we get to that point, that part is already known to work. So a lot of people don't like GMOs. And this is the issue that I've had when I've talked about this before. I've been going to some vegan events talking about this. Most people seem to be fine with it, but I've had a few activists who are not happy with the idea. But I actually would argue that it's not in general GMOs, that there are problems associated with them, but it's not really the science or the organisms for the most part. So this is kind of the equivalence people usually have in their minds when they're anti-GMO, is that Monsanto is evil, Monsanto makes GMOs, GMOs are evil. Well, I'm not going to argue that Monsanto is not evil. They're really profitable, but most of the farmers that they work with are barely making it. Right, so there's obviously some problems there. But there's this, and this is a real problem. Farmers have been sued by Monsanto for trying to save seeds and replant them. This has been the standard practice for all of modern agriculture. So this is not really a problem with GMOs specifically. Traditionally bred strains can also be patented in the same way. People have been sued for illegally planting traditionally bred strains. So this is actually a problem with the patent system. If you want to fix this problem, reform patent law in agriculture. And some of you probably are aware of similar software problems that are out there now. So that I don't really see as a GMO problem. That's a corporate culture and patent problem. People also sometimes don't like the BT gene. This was the first, and actually it was not Monsanto that introduced this. This was the first genetically engineered crop. It comes from this particular bacteria. The interesting thing is that this bacteria, if you eat certified organic food, you're almost certainly eating this bacteria or traces of it because it is sprayed on certified organic food and is perfectly safe for consumption. So you're eating the genes and you're eating its expressed proteins, just like you are in the crops. It's been shown many times to be safe for human consumption and other animals, and it has greatly decreased the use of pesticide on those crops. And there are patent problems around this too, but some people have solved that by, for instance, in India, I don't know if I put this here, in India, people just started pirating it. So they got the benefits and did not have the patent problems, because once enough people pirated it, no one could do anything at that point. So I want to give some examples of good GMOs. So chymosin, I mentioned this before. This is what makes the cheese curd from whole milk, or from the intact casein micelles. Well, it used to be extracted from the stomachs of slaughtered calves. But there were a few problems with this, not ethical problems from the viewpoint of the people doing this, but there were supply problems. And there was reproducibility problems. They would extract this stuff and it wouldn't always work as well. So they didn't have enough. So someone introduced this into E. coli, these genes, and 80% of the world's supply now comes from E. coli. So for those of you who do eat cheese, you're already eating a genetically engineered organism's output enzymes. And this has reduced the reliance on the animals for this. Of course, those animals are still used in other ways at the moment, but this is important for us because if we fully reconstruct the casein micelle, we would also use chymosin. So there is already a non-animal source of it waiting for us, so we don't have to do any of this. The other big one, and there are actually dozens and dozens of medicines, insulin. Insulin used to be extracted from the pancreases, mostly of pigs, but other animals. But it also was unreliable. People would have allergic reactions to some of the other things that didn't get purified out. And so the people that was helping it would sometimes also induce other sickness in. So insulin is now also engineered into microorganisms. And now we don't have to take pig insulin either. We can engineer in human insulin. So humans are using human insulin now. It works a lot better for them and it's very pure. There are also other genetic variations, just changing one amino acid at a time, that have allowed insulin to last longer in the system, act faster, and so it actually has better health outcomes. So there's a lot of really good work genetic engineering for medicines. But there's sometimes problems too. So Roundup Ready is the other one people don't like, and I don't like this one either. There's a few reasons. First of all, this is the only genetically engineered crop ever to be unsafe for an animal's consumption. The Roundup Ready alfalfa was actually taken off the market, I think, a few years ago, because it was actually shown to be toxic. So these things are tested, and there is an example now of it going wrong. But for humans, these crops are all safe to eat. The pesticide glyphosate or Roundup, those traces, it's dangerous stuff, but those traces are small enough that seems to be a fairly well-controlled thing. The regulations seem to work there. That's safe for us to eat. However, the farmers that work with it sometimes do have problems with it. It does actually harm farm workers The other thing it's doing is it's devastating in the Midwest the milkweed populations. And so this is mostly responsible for the monarch butterfly decline, is the use of Roundup. So it's not directly the genetically engineered crop, but that certainly enabled the behavior that is very harmful. So there is an example of something going wrong with genetic engineering. But what I want to argue here is that really you should be against sort of case by case. Things need to be investigated, each one on their own merits. Saying all GMOs is evil, I don't think really helps. And when I brought this up recently, just in the last month or so with people, when I sort of framed it as genetic engineering as a tool, and I've gotten the same response from several people. I didn't realize, I think this is a talking point among anti-GMO people. When I say it's a tool, they say, oh, like guns, right? Those are just tools. Well, I mean, that's true. But really, the only use of a gun is for violence, right? Or a threat of violence, even if it's self-defense. It may be justified depending on what you think of this, but it's always for violence or the threat of violence for any reasonable use of a gun. So I don't like that analogy. I think it's more like metalworking. You can make guns with it, but you can make lots of other things too. So I think that's a much better analogy. You could possibly do really awful things with genetic engineering, but certainly do very constructive things also, like hopefully the examples I showed. So this is another question we get. Are these things going to escape and take over the world? And I'm not sure how many of you are familiar with nanotech, nanoengineering. There was also a similar idea brought up, I think, by this guy Smalley about if we make nanobots to repair ourselves, aren't they just going to turn everything into a gray goo? So that's what I think of here. People ask me this. But we have a lot of defenses against this, actually. For one thing, we use a yeast strain that can't survive on its own. It's uracil deficient. It's missing one of the pieces to make its own DNA. So it can't survive unless it's being grown in this broth that has uracil in it. And there's not a lot of free uracil out in the world outside of these containers. It's mostly tied up in biological systems. So also, do any of you guys know people that have tried to take San Francisco sourdough starter to another part of the country, maybe got a couple loaves out and then it died? Yeast is really location specific. It is evolved very specifically for the place it's in. They don't make it very far. So there's also the problem of horizontal gene transfer. This is if the yeast, a lot of microorganisms, yeast is maybe not as bad as some bacteria or viruses, but they swap their genes all the time. It happens a lot. So we have several ways to prevent that too. First of all, any yeast with a intact uracil pathway in it, we put a poison in for anything that is on its own competent. So no outside yeast should be able to survive in the bath. We also use some other things to kill off bacteria, other microorganisms. But say it did still escape somehow. It's possible that would. There would be no competitive advantage for the organisms having this pathway. They would be using a lot of resources and energy to make cheese protein that really should not help them survive. So this, I think, is a pretty safe thing to be doing. But this is something you have to take seriously. You have to put in these kind of safety protocols because you can let genes escape into the wild. And this has happened with Roundup, that those genes are spreading to weeds and making them resistant. So, but we're aware of this and careful. So I've said a lot of this already, but this is our overall process. We search for the genes of interest in a genome, make the DNA, get it into the yeast. We express and purify the proteins, make the vegan milk, and then make cheese. But you see here the subtitle, we can make cheese from any sequenced mammal. So any sequenced mammal. So cow is the one that everybody expects. This will make cheese most similar to cheese that everybody's used to now. We would like to make human as well, and this kind of freaks some people out, but really this is something that everybody eats at some point in their life, at least milk, and it should have the lowest chance of allergic reaction. Some people are actually allergic to the proteins, not just the sugars in milk. Lactose intolerance is common, but you can also be allergic to the cow proteins. People who are less likely to be allergic to goat or sheep, so you sometimes hear that people drink those. Well, we should have the least allergy to human proteins. And just because we can, we want to do narwhal as well. So we've talked to the Santa Cruz researchers who sequenced the gene. It's not published yet, but they're going to give us the information that we can make the narwhal cheese. And as far as we know, this should be the first narwhal cheese ever made, vegan or not. We don't think there's anybody milking narwhals at the moment for cheese. So what's our actual progress on this? Well, I'll start with some of the soft things, not the technical progress. So we are a functional non-hierarchical organization, basically an anarchist organization. Any of you who have tried to participate in hackerspaces know that this is hard to sustain, but we've had a lot of energy, people are very focused, and we've kept this going for over a year now. We are a nonprofit, so we're incorporated. So we're also getting a lot of engagement from people not with biology backgrounds, which was part of our goals in the first place. And we're doing this with a fraction of the resources available to large labs. And we're doing currently relevant work. So the other thing we're doing is I'm coming to places like this and talking to vegans about the science part, and we're also going to talking to scientists about the impacts of uses of animals and agriculture. Both groups have known a lot more and been a lot more friendly than I expected, so that's been good. We've had really good reception on both sides. So in the lab, what have we actually done? So we've designed 11 plasmids. So we've done the human and cow for the different parts that I said, all those. Actually, humans only have 3 of those 4. The 4 protein subtypes was for cows. And so that doesn't add up to 11. We have a few different variations for some of them. It's codon optimization. You can actually change the code a little bit. It will code for the same protein, but you might get different efficiencies. So we've done some optimization. And then also the kinases, the things that put the phosphorus groups on. So we did this in E. coli first. So this is not to produce protein, but this is to make more plasmid. They become plasmid factories. So once we've got this stuff in the E. coli, we have as much plasmid as we ever need. And we've confirmed that our sequences are good coming back out of the E. coli. We got it into the yeast. Our first attempt, though, something went wrong. We made this yeast and we put it into plates that had no uracil. They should have died and they all grew. So we don't know exactly why, but we just found other people that were willing to donate yeast to us. And the new yeast are working perfectly fine. So it was something with the first strain of yeast. We're working with all donated materials, and we don't have a really high number of resources to verify everything that people give us. So sometimes, and actually anybody who's ever worked in a lab, sometimes things just go wrong. So we think we have the casein proteins coming out. This is based on electrophoresis, so all this tells us is the size of the proteins is right. This past weekend, some people ran the next experiment, which is Western blot, where you take those proteins and then add an antibody to them that will fluoresce if it's the right protein. I actually didn't hear the results of that. I was hoping to tell you guys what happened, but I don't actually know myself. So this is as far as we are. We're pretty confident that we have the right proteins. So the Wired article probably sounded like we were like packaging our first cheese to ship or whatever. We have the protein. We need to scale up now. So I'll tell you a couple other things first, though. We've engaged the FDA. So some people are sort of nervous about biohacker spaces doing this kind of work. Well, we go through the same regulatory process that everybody else does. Now, you could have a problem with that process, and that might be reasonable. But we're subject to the same regulations. And so maybe those regulations need to be improved, but we do go through them. We've talked to a panel at the FDA. They were actually pretty excited about this. They liked the idea. They were fine with it. We should be able to show functional equivalency to things that people eat now, have a very low hurdles to get over to get this stuff out. They did not like the human, though, for exactly the opposite reason that we think it's good. We think it should be low autoimmune response. They think it's going to induce autoimmune diseases. And if that were true, it would obviously be a very bad thing to feed someone. They were basing that on one case where they allowed people to go through clinical trials where they were injecting recombinant human proteins directly into their bloodstream, and that induced some reactions, which sounds more dangerous to me than eating, but it's certainly something to be concerned with. Because of this FDA concern, we're putting the human work on the lowest priority, and we're doing the cow and narwhal first. And then we'll come back and talk to the FDA again later. So the cheese making. So like I said, most of the people in the group aren't vegan. So some people have taken just dried milk casein, regular dairy milk, and started reconstituting it with vegan sugars and fats and making cheese out of it. So I didn't taste this stuff myself. They tell me it's mostly like cheese. So that's as close as I can tell you, is that it seems that if we get this stuff, we can reconstitute it and make cheese. So really the big challenge for us is making the protein and making enough of it. We know the other steps work now. So I'll tell you about the iGEM competition, which is how this group first formed. First thing I'll tell you about it is I'll show you our trophy. So this is the International Genetically Engineered Machines competition. And so the Real Vegan Cheese Project was started as a collaboration between the two labs I mentioned, Counterculture Labs and BioCurious, specifically as an entry into iGEM. We're well past that point now, and we're continuing the project, but this was the competition that we formed around. So this is a worldwide competition in synthetic biology. It used to be all undergrad, but last year they opened it up to community labs. And so we sent, we had 5 people speak, and we won Best Community Lab. We won a gold medal. We were one of 99 gold medals, I think. Gold medal just means you did a good job, not that you were the best. But we also submitted casein plasmids to the BioBrick library. This is a library of standard parts that anybody can use to build their own constructs. And so we now, both on our website, you can find all the information, and also in the BioBrick library, if anybody else wanted to try to do this, they could go do this themselves. So any of you guys could come work in the lab with us, or you can do this in your kitchen. And we started out in a kitchen, actually. Counterculture Labs, or it was a little room off a kitchen. And all this work could be done in that kitchen pretty easily. So past iGEM, what are we doing? So the next steps, working in the bovine casein, we need to figure out if we did the post-translational modification correctly, if we put those phosphorus groups on and the sugars, and then try to figure out if they're really needed or not. So this is the next big step, is identifying what happened to those proteins. So then once we've got that figured out and figured out our strategy, if we need to do more steps on them, we then have to scale up to several grams. We might have micrograms of this stuff. It doesn't take much for analysis. They're in very small amounts right now. We need to scale up to grams, and then we can go reconstruct micelles and make cheese. And that will be the complete proof of concept once we get there. So once we've done that, and probably in parallel, we'll also do all this with the narwhal cheese. And then from there, we need to go to a larger reactor. Part of the Indiegogo campaign I talked about, one of our stretch goals was $10,000 for a 50-liter bioreactor, and we got that goal. So we will have a rather large reactor making cheese. And we just bought I think 5-liter intermediate scale one that we'll start using in the next few weeks. So some of the other output, so part of that Indiegogo is that we had some rewards where you could ask a biohacker and we would make video responses. And a lot of the questions were things that you might expect. One about the pharmaceuticals, which, yes, it's already being done. Why does dairy trigger my migraines? We got one question was, is it more ethical to like robots or dinosaurs? We answered narwhals. I made one video. I looked sort of insane in that video because we had written it and recorded it just in a few minutes. And so I have a screen I'm reading off of that's just off the side of the camera. And so I'm like looking sideways. Yeah, it's not good, but the answer is good. We've had a lot of press. The Wired article is the most recent. We've actually been in a lot of large publications. We did a Reddit AMA. My favorite quote from any of these things, and the O'Reilly Radar blog, was real vegan narwhal cheese. While I'm sure synthetic biology can get weirder, but this is a good start. So I have an idea for the next weirder thing. At the Maker Faire, I talked to some people from NASA who said that some brewers have been sending yeast into low orbit and then retrieving it and making space beer. So I want to make real vegan space narwhal cheese. So that's our next step weirder. Okay, so what about scale up? So the way we're making these proteins is called recombinant proteins. And this will be the first attempt to make a bulk food out of recombinant proteins. No one's done this before. So obviously there'll be some challenges. We think we can get 2.5 grams per liter with the kind of current work that's out there. People have made one of the 4 proteins, kappa casein, in yeast before. So we think this is our first target output. But at this output, 5 grams of protein will cost about the same as 1 liter of craft beer. This is a pretty good comparison because they're using the same species of yeast that we are. We know the output, so we can estimate the cost. At that price, this puts us as a specialty vegan product. This is not a everybody replace cheese product. So we need to get it a lot cheaper there. So we might be able to optimize in the same species, but it turns out that there are other species of yeast that sometimes give higher protein yields. And then there are other fungi. This is so sake making uses the same yeast, but it also uses a different fungus. This species can have particularly high protein output. And one good thing about it is that unlike yeast, it doesn't move itself around. It's not using energy to move around. It stays in one place. And so it appears that not having this motive force, it spends all its energy other places, including making protein. So if we never get the Saccharomyces to give us more protein, there are other things to explore. But you can see from what I've just said that this is definitely a long-term project, right? Especially, like I said, we're working nights and weekends on this. So I would say for an all-volunteer group, we've made good progress. This is certainly slower than an academic lab would do on this. But we also are doing it for much less money, right? We don't pay anybody's salaries, so that's the trade-off we're making. So if we succeed, at the 2.5 grams per liter yield, the cheese would have about half the greenhouse gas impact as dairy. This will only improve as we improve yield. So we could significantly reduce the greenhouse gas impact. I tried and did not really come up with any water calculations I believed were exact, but it looks like it will definitely be less. I'm not really sure how much less. If we never get the price down, but there's some really good vegan cheese that's expensive, it may turn a few people vegan. I'm not sure. But the real payoff for me is if we can make this cost competitive with dairy cheese, and then people basically go vegan without deciding to go vegan. One of our members really wants to see this. Right now we're doing all this bioreactor stuff where we're keeping other species out, containing it very carefully. One of our members really likes this idea of everybody having a little pot in their kitchen that's constantly churning out cheese proteins. And then they're never paying anybody for cheese. They just take like ladles out of this and cook with it. So yeah, this would be further down the line. Maybe someday we'll have our own home vegan cheese factories. It's possible. So some spin-offs of this project. Some people in the group are, and these will be separate projects, this won't be part of vegan cheese, but some people in the group are trying to do a cheese biome project, just like there's a human gut microbiome and soil biome projects going on now. These people want to take samples of all the cheeses around the world that they can find and characterize exactly what species are living on them. If we characterize all these, we can try all of them on vegan media and maybe make more interesting vegan cheeses. Along the same lines, they're collecting, or we're going to start collecting, all these different cultures and transferring them from the dairy media that they're grown on now into vegan media, so vegan sugars, and grow them up. And then after a few generations, there won't be any animal products left. So this, some people might be uncomfortable with, but if you eat vegan yogurt now, those cultures a few generations ago were grown on dairy milk. There's no way around it. That's the only way that these particular strains have been grown for probably hundreds of years, if not longer. So this is how that's being done. The vegan cultured nut cheeses that are out there also, I've talked to Miyoko and some of the other makers. When they buy them from their suppliers, they are vegan when they get them. But I'm sure those suppliers did the same thing, that they switched them to a vegan media, and that they were grown on dairy at some point in the recent history. So we're trying to make a large library of these same kinds of cultures. So, there's a lot of this kind of stuff going on right now, actually. This is the one that's most interesting to us because they're doing the same thing we are. They are a venture-backed company, pretty well funded, I think about $2.5 million. They, of course, are full-time employees, and I think they're trying to do the whole thing. I think they're trying to do the proteins and the fats and the sugar, in which case they should get something identical. They should really get milk. I think this is a harder goal, but then they have more resources, so maybe they'll do it. The only one point that we may be rivals with them, I would love to see them succeed, but we are open sourcing everything so that as many people as possible can try this out themselves and try to make a working product. They are patenting things, I think. They haven't told me this, but they're a venture-backed company. I assume they're patenting. These other companies, actually, this has been in the news a little bit. You guys may or may not have heard of Impossible Foods. They are making fake meat that is, actually, I've tried it. It was too realistic for me. I couldn't eat it. They're taking plant proteins, making the right textures like other people do, but they're also taking hemoglobin-like proteins from legumes, and they found that when they cook plant proteins with this hemoglobin-like protein, they make the exact same small molecule flavors that you get from real meat. So the flavor molecules are the same, so it tastes really like meat. This was started by Pat Brown from Stanford. He's a pretty well-respected professor and invented the DNA microarray. He left Stanford to start this company. So that's pretty cool. In Hampton Creek, I'm sure you've all heard of. They are by far the most successful people in this space right now. They're making them fake eggs. They have a sort of different strategy in that there's some genetic engineering going on at Impossible. Hampton Creek is staying away from that. And I think it's mostly because of the perception of GM foods. They did a kind of convergent evolutionary analysis. They looked for the closest plant proteins in evolutionary history to the egg proteins. They found one particular kind of like yellow pea or something that had a very similar protein, then figured out the processing to make something very much like eggs. And there's a lot more along these lines. So I think there's sort of a moment in like high-tech vegan food right now. So if you guys think this is an interesting project but would like to see it move along faster, the best way is to show up at our meetings, especially show up in lab. Especially show up in lab if you have any previous lab experience. So we meet 7 p.m. Mondays at Counterculture Labs, except for the first Monday of the month at BioCurious in Sunnyvale. But any of those you can join. We use Zoom, not some of the other better-known ones. I'm not sure who chose that. It works well. Lab work goes on weekends at BioCurious. Some at Counterculture Labs, but that's why we're running the Kickstarter. We need to do electrical and plumbing upgrades to really do much lab work. We have a mailing list. I put my contact information up there also. We also, again, I already mentioned, but we have a Kickstarter going on. So please contribute if you can. And I think that's it. Yeah.
Question, Mike. Cool. Does anyone have any questions?
So what determines the cost of the 1 liter of yeast or whatever? Like, do the yeast keep going forever, and is that just sugar, or?
Yeah, but we'll probably have to periodically kill off the yeast and grow up a new batch. The purification process, it's basically you feed sugar in, you feed them, it takes some fresh water every once in a while. Get these proteins out and you have to filter it. Just looking at the cost of like people's time running the reactor and the sugar, and then the output yield is most of the determination of that cost.
So just curious, looking way down the road, you said that you've incorporated as a nonprofit. What would you do once you made cheese? Would you be a nonprofit and sell cheese and invest the profits back in more science, or do you have a plan?
That's not particularly well worked out yet, what our model is at that point. We probably would try to sell cheese ourselves. I think some members of the group would like to spin out as a startup. And anybody could do this now. I don't think our technology is quite far enough along for someone to take it and use it, but this is completely open. There are no restrictions on use at all. So if someone else wants to profit from this, that's fine. And some members from the group might go do this. And hopefully we would do it better since we were the ones who invented it, but not necessarily. So we don't exactly have a business model. We have a model for getting the technology ready, and then we'll figure it out at that point, I think, is the actual answer.
What's your rough estimate of timeline to when you might get to that point?
So last October at the iGEM competition, we were telling people about now we would have tasteable amounts of the protein. At that point, we hadn't shown that we even thought we had the right proteins. Okay, I'm not sure if... so we've gotten further. We definitely have made some progress. Really, from where we are now, having a first tasteable cheese, if we worked really efficiently, it could be, really a professional lab should be weeks to a month out, so we're probably 3 or 4 months out at least, maybe 6. Then the first scale up will be maybe another 6 months past that. So we may be a year out from scaling up to large enough amounts. I think this year someone will taste the cheese. Now, we can't legally give it to anybody outside the group to taste. We need FDA approval before, and we're not even sure about tasting it ourselves. We may not really legally even be able to taste it ourselves. But the regulatory approval, as slowly as we move in the lab, regulators are probably slower. I'm going to play this video real quick, and then we will go back to questions. It's still going from my laptop. Yeah, sorry about that. Well, maybe I can just mic my laptop. Hopefully not get horrible feedback.
To make cheese, you need milk. And to make milk, you need mammals.
I'm not sure where my...
farm animals aren't always treated as well as we'd like. Then there's the environmental impact of having 260 million dairy cows worldwide. And let's not forget that 60% of adults are lactose intolerant and can't eat many fresh cheeses.
Sorry about that. Do you...
Yeah, you want this? I just cranked up the volume on it.
Okay.
Let's try it again. I like your mic hack.
I didn't quite figure out where the speakers are in this section.
To make cheese, you need milk, and to make milk, you need mammals. But as we all know, farm animals aren't always treated as well as we'd like. Then there's the environmental impact of having 260 million dairy cows worldwide. And let's not forget that 60% of adults are lactose intolerant and can't eat many fresh cheeses. What if there was another way to make cheese without the problems? We're a team of citizen scientists and biohackers based in the San Francisco Bay Area, and we're working hard to reinvent how cheese is made. The big problems with cheese all have to do with where we get the milk, so we're going to replace cows with these little fellas. This is Saccharomyces cerevisiae, better known as baker's yeast. We're going to give baker's yeast the ability to make milk. Okay, maybe not quite milk, but at least milk protein, which is the important part of milk when it comes to cheese. Here's how it works. We're taking genetic sequence for milk protein from cow DNA and creating cheese protein genes that function in yeast. We then insert the cheese protein genes into living yeast cells. The genetically modified yeast will produce cheese protein as it grows, and raw cheese protein can then be extracted and separated from the yeast. To make real vegan cheese, we simply take the cheese protein and combine it with the 3 other main ingredients of cheese: fat, or in this case vegetable butter, lactose, or for our cheese a vegan sugar, and good old-fashioned H2O. Mixing these up makes a kind of vegan milk, and from this milk we'll use the same cheese-making process that has been practiced for thousands of years to make Gouda, Emmental, and even Parmesan. Our team includes experienced biotech engineers, computer scientists, and dedicated vegans. As with all scientific research, we can't be completely certain that this will work until we try it, but with your help, we're confident that we will be able to make vegan cheese protein and with a lot of hard work, turn it into the world's first real vegan cheese.
Okay, is this, can you turn the volume down a little more? Okay, any more questions? Yeah.
So part of flavor is salty or savory or whatever like that, but a lot of it is small molecules that go through your nose and are sensed. Do you have any concerns about there not being other small molecules that would contribute to the flavor of the cheese?
So most of those flavor molecules are actually, can you turn it down more? Most of them are made by the microorganisms that are grown on the cheese, at least for aged cheeses. And so those organisms should make the same flavors grown on our cheese. There's one danger there. Some of that, what they eat is the lactose. And so we're using different sugars. Most of the same molecules will still be made, but there may be some differences. So there is actually a chance that we'll get some different flavors. We're not sure about that yet.
So they eat both the proteins and the lactose and create the small molecules.
Yeah, they don't eat the proteins that much, actually. So that was the fake meat stuff from Impossible. It was reactions with the proteins. For the most part in cheese, they're not eating the proteins. That first enzyme cleaves the water-soluble parts off. But after that, it's mostly eating the sugars that makes the other flavors. Yeah.
Did you get a chance to test the functional properties of the casein that you already extracted?
So the small amounts we've had are not reconstituted into the casein micelles yet, and that's where it's most important, and we've not gotten that far yet. So that'll be the biggest test of our technology.
Because you could extract something that's like chemically like casein, but it won't work like casein.
Right. If we get something that's chemically identical to casein, it's already been shown that you can reconstruct the individual parts and make the micelle. But if we get something that's very much like it but not identical, yes, there's a chance for failure at that point. And the two biggest problems, we really should get the same protein sequence. Really where the phosphates go on in this translational step later, and where the glucose goes on that hydrophilic tail, those are the biggest chances for failure we have. And that part we just don't know yet.
And you have been asked this question, but I didn't get the answer. What's roughly like the cost of the product like right now?
Right now, well, we think once we get to the estimated 2.5 grams per liter output of the protein, for 5 grams, it could be $10 to $15. That would be enough protein to make 1 to 2 servings of cheese, so it's going to be very expensive at first. It will be, right now, probably on par, maybe even a little more expensive than like Miyoko's cheeses or Kite Hill cheeses. We should be able to beat that, though. I don't think for them that most of their cost is the cost of cashews, and that's not going to come down much anytime soon. It'll probably go up. If we can engineer our stuff to give higher outputs, we should be able to get to lower cost. But getting to a low-cost alternative to current cheeses is a years-long project. This is not, we're not there at the moment.
Curious to know why it is called real vegan cheese. I mean,
Right. Yeah. So.
is it real, and is it like
Yeah.
other vegan cheese are not real, or
Right.
this is the real cheese?
Yeah. We call it that because it is real cheese in that the proteins are the same proteins that you get in dairy cheese. So actually, legally defined, our stuff is still not cheese. The legal definition of cheese is not just milk, but it's milk expressed from the udder of an animal. So our stuff, even if it's molecularly identical, will not legally be cheese. However, we felt that it was close enough that for this project at least, we couldn't sell it with that name. But for this project, it was sort of a clever, catchy name. We were trying to raise money on Indiegogo. But it's real because it is the same proteins that are in dairy milk, but it's vegan because no animal is actually used in the production. So that's where that name came from.
Nobody can sue you even if you call it like cheese.
Right. So, yeah, we can call it cheese when we're making it in a lab, but we can't legally sell it under the name cheese because it was not expressed from an animal.
Thanks.
I was interested in what you were saying about the process of rennet and the important component, I forget the name of
Chymosin,
it.
yeah.
Chymosin, coming from non-animal sources, about 80% now. So I like the way that means that we're maybe all going to meet in the middle, roughly. But my understanding was a little bit crude. I always thought that rennet was the part that came from the calf's stomach. And so if that important component, what else is there to rennet, and how is that?
I don't actually know what the other components are. So rennet is a complex of a bunch of enzymes that travel together. The function of those other enzymes, I'm not sure what they are. Chymosin is the one that was singled out by the large cheese manufacturers, people like Kraft, as this is the one we need. But as far as the function of the other enzymes, I don't actually know.
So will we be seeing a lot more claims of rennetless cheese? Do you anticipate?
No, it's now you'll see it called plant rennet or microbial rennet. Those are recombinant proteins. So it's labeled now. If it just says rennet, it could be any of these. Since on a molecular level it is the same, the FDA in this case did not make people label it as something different. So if you just see rennet, you don't know if it was animal or microbial. But if you see plant or microbial rennet printed, then it came from a genetically engineered source.
Thanks.
You talked a little about at the beginning about casein and its functional properties, how like the hydrophilic parts go on the outside and the hydrophobic parts take the fat around, but all proteins have hydrophilic and hydrophobic parts. Can you talk a little more on the technical side about what makes casein special and why you specifically need casein to make cheese?
Well, it's this kind of combination of the size of this micelle it makes. Nothing else makes a micelle quite like this. Plus this cleavable part that allows you to transport in liquid form and then cross-link it in the 3D network with the chymosin. Most proteins don't form these very large. These are microns across, so most proteins don't form these large clusters. So mostly they're folded up very small, they're nanometers across. To give these macro physical properties of milk, you need these micron size clusters. That just doesn't happen in other proteins, at least not these ones that are as water soluble. There could be, there's still a chance that someone will discover a plant protein that works like this, and this is not needed because there's plenty of this plant protein. That is perfectly possible. And it's possible that activists will convince everybody to go vegan before we finish this project, which would be great. So those things are perfectly possible. I can't tell you that there's nothing else that does this. We don't know of anything else that on this size makes these this large of a micelle that stays suspended. And also the calcium carrying property seems to be important for this structure. This is the hard cross-links that make it pop back into place. So these calcium nanoclusters, when you stretch it, it's not actually the links between the individual micelles, those kind of all smear together. It seems to be those calcium nanoclusters that are the hard links that give it the stretching and then springing back. And there are not other proteins, as far as I know, that have those metal nanoclusters in them.
So I know there's a lot of concerns about the health of dairy products. Could you speak about that?
So I actually don't know. It's possible that there's I have people, my mom included. My mom is vegan for her own health. And I don't want to argue against her too much because I want her to keep being vegan, but I don't actually have any good sense. I've tried to read up on this, and the scientific literature is kind of sparse on this, and it's hard to know what to believe. There's not anything that's really that conclusive. So, really, I don't know. My argument is that, again, it would be great if activists could convince people to go vegan for animal rights reasons. If it's unhealthy, it would be great to get people to stop doing it because it's unhealthy as well. But I also think with your own health, people should be able to make their own choices. The impacts on others, we need some social regulation around maybe, but for your own health, if you know it's unhealthy, but you're going to do it to yourself anyway, maybe it's not great, but I'm not going to go out of my way to stop you. So hopefully someone will figure out the truth of whether milk is actually harmful or not, and that we will all have that information available. But if people want to eat that protein, if I can make a vegan way for them to do it, I will.
Is that it for questions? Okay, I just have one. So the narwhal, when I first heard about you guys, I thought the narwhal cheese was amazing. And first of all, I thought they were extinct already. So I was like, yes, you can make cheese out of extinct animals. But can you?
If the genome is sequenced, which I think there are a few that are.
Mammoths, maybe?
There might be mammoth. We might be able to make mammoth cheese. Let's see, what else?
What's the minimum amount of material, or what kind of material do you need to sequence?
So saliva works. Skin cells, I believe, work. Part of the problem is that in samples that are old that still have some soft tissue intact, the DNA can still be heavily degraded. Just because you have that tissue doesn't mean there's intact DNA. So I don't know what extinct creatures actually have a full genome that have been sequenced. But we should look into that. Mammoth cheese would be
Yeah. It's on the roadmap, right?
Yeah.
All right. Well, thanks, Ben. Really appreciate your time, and thanks everybody for coming. We still have about 40 minutes.